Hello everyone, my name is Prajakta Dhage and I am from Department of Zoology, KTHM College, Nashik. Today I'll be taking a class for MSc Part 2 and the semester is 3. Subject that we are dealing with right now is Insect Physiology and chapter is 17th chapter. Today's topic is going to be endocrine organs and this is the second lecture of this particular chapter. So, in the initial part or in the initial lectures, we have already seen the types of hormones that are present in insects. Today, we'll be focusing on the endocrine glands in insects. Okay, so this is the first part of this particular lecture. Let's begin with the glands. Now, what we'll be talking about today is the glands that produce ictiosteroids. Okay, so in immature stages of all the insects, moulting hormones are produced by prothoracic glands. In adult females, where the same hormones are produced to regulate embryonic development, the follicle cells in the ovary are principal source. Although there is evidence that some ictiosteroids are produced elsewhere in the abdomen of insects, possibly by the oonocytes. Now let's move on to the first gland, which is prothoracic gland. In this particular image, I have encircled this particular gland with red color. Please have a look at the diagram. So, the prothoracic or thoracic glands are a pair of usually diffuse glands at the back of the head or in the thorax in most of the insects. But in the cases of but in the base of the labium in Thysanura. Each gland has a rich tracheal supply and often a nerve supply. In most insects, the nervous connection is with the subesophageal ganglion, but in some, there is also a connection to the prothoracic ganglion, and in cockroaches, there is connection to the brain. The prothoracic glands of Heteroptera have no nerve supply. The glandular cells making up the gland is most hemimetabolous insects and in Coleoptera are small, up to 25 micron across, and they are present in large number and undergo cycles of mitosis. However, in most other holometabolous insects and in Heteroptera, the cells are large and they are present in relatively small numbers, ranging from about 30 in the larva of Ditria to 250 in Hylophora. The cells in these species do not divide but undergo endomitotic division so that they become polyploid, increasing greatly in size through the larval life. The gland shows cycles of development associated with secretion. The prothoracic glands degenerate in the adults of nearly all insects, but they persist in Pterygota, which continue to mold throughout their life, and in the adults of Solitarius locusts. Normal persistence of the glands throughout the development results from the presence of juvenile hormone in the hemolymph. Their breakdown after adult emergence is due to lack of this hormone. So this was about the prothoracic gland. Let's move on to another gland which is called as Corpora alata. Again in the red marking you can see here this particular structure is nothing but the corpus alatum or Corpora alatum, or the Corpora alata are glandular bodies, usually one on either side of the esophagus. Although they may be fused to form a single median organ as in higher Diptera, each is connected with the corpus cardiacum on the same site by a nerve carrying fibers from neurosecretory cells of the brain. In addition, a fine nerve connects each corpus allatum with esophageal ganglion. This is a major nerve in Ephemeroptera, where the nerve from the corpus cardiacum is absent. In Thysanura, the corpora alata are in the base of the maxillae. In addition to a fine nerve direct from the subesophageal ganglion, they are innervated by the branches from the mandibular and maxillary roots of the subesophageal ganglion. In Thysanura and Phasmida, the corpora alata are hollow balls of cells with gland cells forming the walls. Elsewhere, they are solid organs of glandular secretory cells, often with the lacunae between the cells. In hemimetabolous insects, the gland contains large number of small glandular cells. By contrast, in holometabolous species, the glands are large and few in number. There are only about 20 in Drosophila and some other beetles. The corpora alata produce juvenile hormone whose principal function are regulating metamorphosis and in some species yolk synthesis and deposition in the oocytes of the adults. Clear with the corpora alata, singular corpus allatum and plural is corpora alata. Now let's move on to corpora cardiaca. The corpora cardiaca are a pair of organs often closely associated with the aorta and forming part of its walls. In higher groups such as Lepidoptera, Coleoptera and some Diptera, they become separated from the aorta. Corpora cardiaca are not known to be present in Columbola. Each organ contains the endings of axons from the neurosecretory cells in the brain and other axons passing through to the corpora alata. In addition, they contain intrinsic secretory cells with long cytoplasmic projections extending towards the periphery of the organ. These cells are probably derived from neurosecretory cells, rather than being glandular cells with a separate origin. Their projections probably facilitate the release of secretions into hemolymph. The corpora cardiaca store and release hormones from the neurosecretory cells of the brain, to which they are connected by one or two pairs of nerves. They serve as neurohemal organs for several different hormones. In addition, the intrinsic secretory cells produce adipokinetic hormone and some other peptides whose functions are often unknown. Sometimes storage and secretory cells are intermingled. Now let's move on to another type of gland which is called as ring gland. Now the ring gland of Cycloraphus diptera. In the larvae of Cycloraphus diptera, the ring gland surrounds the aorta just above the brain. It is formed from corpora alata, corpora cardiaca and prothoracic glands all fused together. Although the component element can still be identified. The ring gland is connected to the brain by a pair of nerves. It also has a connection with recurrent nerve. The larvae of Nematocera have completely separate endocrine glands, but larval orthorhapha approach the cycloraphan condition except the corpus alatum tends to be separate from rest of the single median lobe. Now let's move on to this was about the ring gland. Now let's move on to another type of endocrine cells which are situated in the midgut. All the insects have cells in the midgut epithelium, which by virtue of their ultrastructure and immunocytological properties are believed to be endocrine cells, although in no case has their endocrine function been proved. These are isolated cells scattered amongst the principal midgut cells. They stand on the basal lamina of the midgut and are therefore in direct contact with the hemolymph. The basal plasma membrane is flat, not deeply infolded like that of principal midgut cells, and they release their contents into the hemolymph by exocytosis. The cells are of two morphological types: those that extend to the lumen of the midgut by a slender process, and those that apparently do not. These are called open and closed cells respectively. Where the open cells reach the lumen, they have microvilli. It is probable that endocrine cells which extend to the gut lumen respond directly to the substances in the lumen. Immunocytological studies indicate that these cells produce peptides. The adult female mosquito Aedes has about 500 endocrine cells scattered throughout the midgut and a ring of cells in the pyloric region. By contrast, at least 30,000 endocrine cells are present in the midgut epithelium of Periplanata. The endocrine cells in the midgut of Aedes are immunoreactive to 4 different antisera, each cell containing only one peptide. These are perhaps the only peptides present in the midgut cells of the Aedes, but other insects are known to have as many as 10 different peptides in the midgut endocrine cells. Although the function of these peptides are not known, they are presumed to have hormonal function relating to digestion and absorption, perhaps regulating the synthesis of digestive enzymes and post-feeding diuresis. Some of the peptides are similar to some peptide hormones secreted by endocrine cells in vertebrate gut. For example, half of the 500 endocrine cells in the midgut of Aedes are recognized by an antiserum to the peptide phenylalanine methionine arginine phenylalanine amide FMRF amide. All the cells exhibiting this response are situated in the midgut where the blood meal is digested. Other cells may have different functions, perhaps including the regulation of gut motility. The midgut epithelia of several caterpillars have been shown to contain peptides that induce contraction in visceral muscles. Now let's move on to another gland. This was all about endocrine cells of the midgut. Now let's move on to the last part of this particular lecture, which is epitracheal gland. Epitracheal gland are also known only from Lepidoptera, although it is likely that they will be found in other insects. In Mandioca, each epitracheal gland is a group of 3 or 4 gland cells attached to a trachea near each spiracle. One of the cells increases in size prior to ecdysis, reaching a diameter up to 250 microns and regressing again after ecdysis. These cells are the source of ecdysis triggering hormone. And this was all from my side about today's lecture, and thank you so much.